Salmonella enterica is a major foodborne pathogen, particularly associated with the poultry industry, and has over 2,600 serovars that differ in geographical prevalence, transmission route, and pathogenic potential. Current surveillance protocols rely on culture isolation and traditional serotyping, which often fail to detect less abundant serovars, causing an underestimation of serovar prevalence. We have evaluated a new metabarcoding method called CRISPR-Sero-Seq to detect multiple Salmonella serovars in a single test. We applied this method to uncover Salmonella serovar prevalence in Ontario's poultry production environments. The metabarcoding method involved a few steps. A sample was first enriched overnight. Genomic DNA was then isolated from the enriched sample. The DNA was used as a template in a subsequent PCR reaction, where Salmonella CRISPR spacers were amplified to produce sufficient serovar-specific sequences. These sequences were then tagged with a unique index for each sample. The tagged PCR products were sequenced via a high-throughput sequencer to generate sequence reads. The sequence reads were analyzed against a Salmonella spacer database to call a serovar. The spacer sequence frequency was also used to estimate the relative abundance of serovars in a single sample. We first evaluated the method using pure cultures and spike samples. The method successfully detected the 34 most commonly isolated Salmonella serovars, confirming the method's specificity. To confirm the method's sensitivity, we spiked 15 target serovars into 15 Salmonella-negative environmental samples at 10 to the 4 CFU per mil, each with 2 other distinct background serovars at 10 to the 5 CFU per mil. In each of the 15 samples, the target serovar was successfully detected, along with the 2 background serovars. Furthermore, similar read frequencies were observed within triplicate samples, suggesting a high level of reproducibility. We then applied the method to 305 naturally contaminated Salmonella-positive environmental samples from 160 farms. We identified 29 distinct serovars, with Infantis, Kiambu, Brinderup, Kentucky, and Schwarzengrund being most prevalent. We also found 57% of samples contained 2 to 6 serovars in a single sample. This is significant as the traditional culture method can only detect the most dominant serovar in each sample, meaning that up to 5 serovars can potentially be missed in samples that contain multiple serovars. 70 samples were further analyzed using traditional serotyping, revealing 92.8% agreement between the new metabarcoding method and the traditional culture-based method in detecting the most dominant serovar in a sample. Overall, the metabarcoding approach was effective in detecting multiple Salmonella serovars in poultry environmental samples. This is important to the food industry because the validated method can be used to improve poultry surveillance programs and interventions by detecting and monitoring all serovars in poultry farm environments. Moreover, this method can be used as a rapid tool in foodborne outbreak investigations where multiple serovars may be involved.